Today we have a very special episode coming for you. It's all about forensics. It's kind of cool actually. This episode is basically to raise awareness so that we can get money and donate it to scientific research. We need to develop new technology in order to help people who were wrongfully convicted before, which I have a special surprise for you at the end of this show. First, we have Professor Nazly coming in and talking to us about STR, which I don't really know what that is, PCR, gel electro something, and then we have an FBI agent coming to talk about this new program called CODIS. And lastly but not least, we have a very own wrongfully convicted person coming in and talking to you guys about her case. Lastly, we have a wrongfully convicted person coming in to talk to us all about her case, which is very interesting. Now help me welcome Professor Nazly. Hello, Professor Nazly. It's a pleasure to have you here today with us.
The pleasure is all mine.
Okay, Professor Nazly, could you please explain to us what STR means and what it stands for?
Sure. Okay, so STR stands for short tandem repeat. They are the stretches of DNA that are repetitive. For example, AGAT, 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 AGAT.
A short tandem repeat, or STR, is a region of DNA composed of a short sequence of nucleotides repeated many times. Since the number of repeated sequences in a given STR varies from person to person, pinpointing these variants can be useful in DNA fingerprinting. As the name implies, the repeated stretches in STRs are short, only 2 to 10 base pairs long. For this reason, and because they are dispersed more evenly throughout the genome than the longer variable number tandem repeats, or VNTRs, STRs are favored by forensic labs. Large databases of information on STRs in the general population tell analysts how much variation exists at any given STR location. That information can help forensic analysts determine the conclusiveness of a match between two samples. If 10% of people have the same number of repeats at a given STR, for example, finding a match at that site is merely suggestive that two samples are a match. If two sites with the same prevalence match, the odds drop to 1 in 100 that the similarity is coincidental. If 13 sites match, the odds that any two people would possess such a fingerprint are so small, about 1 in 10 trillion, that the result can be considered a definitive match.
Professor, can you please tell us what PCR is?
Okay, so PCR is a polymerase chain reaction, which is a copy of million DNAs from a few cells. Basically, it acts like a photocopy.
Can you please explain to us how PCR and STR are related?
PCR and STR are related because they both have the repeat of DNA from each person. Okay, so there are some pictures and videos to explain.
Can you please explain to us what gel electrophoresis is?
Of course. Okay, so gel electrophoresis is a technique used to separate mixtures like DNA and proteins. The separation is based on how positively or how negatively charged a molecule is and its size. So what you need is an nylon membrane, running buffer, dye, gel, samples, and stand. First, you add the buffer into the gel box. Then you add the gel, which will be used in the running buffer in the gel box. And then you add the loading due to all the samples. And then you add the dye, loading dye to all the samples. A little amount of samples and standards are added to the well in the gel. And then you connect to a power source. This is the last thing. And there are some pictures to prove it.
Thank you so much, Professor Nazly, for your time here.
It's all my pleasure.
Okay, so sadly, my FBI friend couldn't make it here today, but luckily he had filled me in on all the things that I need to talk to you guys about. So, First of all, they created this new program in 1998 called CODIS. CODIS stands for Combined DNA Index System. And what this does is it has a complete, it's a software of DNA database that establishes all law enforcers a criminal justice DNA database so that it can help speed up the process of finding victims. The reason CODIS was created is after the DNA Identification Act in 1994 had asked them to create such a program. The function of CODIS is speeding up the process of finding victims with any small sample of DNA. People generally ask how successful CODIS is, and you all might think that it's so successful and everything, but it's not as successful as you might think. Due to the amount of people who are related and share similar chromosomes and similar patterns, it is an efficient program and everything, but sometimes having these samples isn't enough to find exactly who you're looking for. There are major risks that CODIS has, and the two major ones is that people can easily now fake their DNA samples, and so that can lead to DNA fraud and everything. And the other thing is that another risk would be it's an invasion of privacy to know that the FBI and any kind of law enforcer has your DNA, because your DNA is something that's very, like, it's just yours, and for them to have it will lead to a lot of problems, and it will lead to discrimination. And now I'd like to welcome our special guest for tonight's show, the exoneree, Kathy Gonzalez. Well, let's get started. Kathy, I think I speak for everyone here that we'd all really want to know your story and what happened to you.
I, Kathy Gonzalez, was wrongfully accused and convicted at the age of 24 with the murder of Helen Wilson, my neighbor. I lived in the same building as Miss Wilson, and that was probably why they thought I murdered her. Dean, Sheldon, and I were sentenced to 10 years in prison. I served 4.5 years until they finally exonerated me.
I just have a question. What were you convicted with?
I was convicted with murder, and the person who was in charge of my conviction was a forensic technician named Joyce Gilchrist, and she was the reason why we were all wrongfully convicted. She was the reason that Smith, who actually committed the crime, was...
Wait, go.
Why were you, what were you convicted of?
I was convicted of murder, and the person behind this was a forensic technician named Joyce Gilchrist, and she was the reason why we were all wrongfully convicted. She was also the reason that Smith, who actually committed the crime, was not called from the beginning of the case.
Joyce Gilchrist was known to be one of the biggest forensic science technicians frauds in the US, just so you know.
Yeah, that's probably why
Continue.
this happened. I testified at White's trial that he had talked to her about committing a burglary, and after everything was over, I was rewarded with $350,000 for being wrongfully convicted. So it was a good ending.
How was the use of forensic science used in helping to exonerate you?
Forensics was used to help exonerate 3 people in this case, not only me. The prosecutors were able to obtain semen from the crime scene, and the results of the DNA test matched Bruce Allen Smith, which was the person who actually murdered and sexually assaulted Miss Wilson.
Well, thank you very much, Kathy, for being here today and sharing your resourceful story with us. And that's it for the show. Thank you.
Thank you.